This is Radio EBM.
Yeah, so this assay seems to be quite complicated. What do you consider to be the most critical parameter to monitor for maintaining long-term stability and accuracy for this assay across the labs?
Right. So mostly because, for example, because we also do with students, undergrads, we develop some other methods and with a research purpose. Most of the time, if you publish it, eventually student leave and the project finished, and then what you do, you never run after that method, So you mostly of the time in the practical mass spectrometry world, like a research, we're not running for 5 years, we just switch into another this is what we did with a glycated albumin. We developed mass spectrometry method, but we're not running routinely. But if you need as a reference method, it's like a different complexity, It's more complicated because you need to run for 10, 15 years. For example, C-peptide, we already running 15 years, and you should be very confident that you're not drifting, because it can be recalibration purposes, can be other obstacles like you switching mass spectrometer, you buy another platform, and then eventually it can like a drift eventually or quantifying results for calibrators. And why we do this, and your question, how we keep in this stability, long-term stability, is kind of, again, very niche of the niche because nobody usually does it for a long time. And for that purpose, we're running clinical assays that we know that is stable. We do always compare them to our reference method. And this paper that we publish, it's an extra method that we can be sure that our methods not drift, our reference method not drifting, So kind of this, you need to have extra, several legs eventually to keep it consistent. It's very important in this world. 1 gram, it's 1 gram after 100 years the same.
So it's not just one thing. You need to pay attention to the whole bunch of them.
And this, I would say, this is most complicated part, the most difficult part to keep it long term. Develop it can be okay, you can develop, but how long you can run it, this method in your lab, it's kind of this is more complicated kind of.